Okay, so the next thing, here we have the transfer RNA. It looks kind of like a snake or an ampersand, like ampersand is like a plus, symbol for plus. Transfer RNA has two things on it. It has an anti-codon, which is this. Remember, these are the three bases that are complement codon. Codon is in the messenger RNA. Anti-codon is in the transfer RNA. The transfer RNA also carries an amino acid. So at the end, I'm just going to run the tail. We have an amino acid. It's specific to this anti-codon. And how do we know which one it will have, which amino acid it's going to carry? We have to use the genetic code. So we have to look at the first letter, and I'll show you that in 1 minute. So here we have amino acid, and in this case, the amino acid is isoleucine. Okay, you don't have to memorize this. This is just, that is the amino acid that is specific to this anti-codon. So the anti-codons pair up with specific amino acids. Okay, so this is going to carry this. Now what happens is now these transfer RNAs are floating around. They come to the ribosome. They float around the cytoplasm. They come to the ribosome and they land. Ribosome. And they match up their anti-codons with the codon of the messenger RNA. So here we're going to take this transfer RNA. I'm just going to move it up here. We're going to match up A, U, C. Okay, so this is and it has an amino acid. In this case, it's isoleucine because that's the one that's specific toward this anti-codon. So now they've matched up. We have an amino acid now. So this is the first one here. Normally, you're going to have a start message to start this process. However, right now we have created an amino acid. That's the first one. This process now is called translation. So translation is when you're going to translate. If you're translating from one language to another, which is really what's happening here. We're translating from the genetic code language, which is made up of these bases, 3-letter words, codons, anti-codons, and we're translating it to the language of amino acids or proteins. So we are really going from messenger RNA to transfer RNA. We're going from genetic code to protein code. So genetic code going to protein code. This is the process called translation. And this happens in the ribosomes. This is where it happens. So the ribosome right here. Okay. This is the big ribosome right here. The ribosome really has a couple parts. We're not going to worry about that. So this is where translation happens. So we're going from genetic code language to protein code. Right now we've made one amino acid. Remember, proteins are made of amino acids. So if we get a bunch of these and put them together, we get a protein. Right? So this kind of works like an assembly line. This one, we're done. This codon matches with this anti-codon. Now we're moving on to that next codon. So CUA is going to have an anti-codon of G, A, U. So we're going to have a different anti-codon because we have a different codon. We're going to have another amino acid, another transfer RNA, anti-codon matches with a different amino acid, aspartic acid. You don't have to memorize the names of the amino acids. It's just a different one because it has different anti-codon. So this is what happens. A new transfer RNA comes in, matches up, and it's going to, as it comes in, this amino acid from the first transfer RNA is going to be picked up by the next transfer RNA. So now we have 2 amino acids. And it's kind of like an assembly line. Then we have the next codon is red. We have another transfer RNA coming in with the anti-codon and a different amino acid. And then that third one comes in and it picks up these 2. And you can see now how we're building we're putting these amino acids together and making a protein. So this is how it's done. This is what's happening. It's hard to even imagine the engineering of nature because this is happening in your cells all the time. Your cells, you make proteins. There's proteins that you can make in your body. You need. And this synthesis or making of proteins is the process of translation. So translation, remember, translating from the genetic code to the protein code, it's really protein synthesis. Okay, so translation. Lots of enzymes. Remember, enzymes are chemicals that speed up the reactions. It's just like I said, it's just these amino acids keep combining, and eventually, once you get a protein, you come to a message that says stop. There's anti-codons that actually indicate stop. I mean, this is unbelievable. How does nature know how to do this? That's when the protein is going to be released and sent to wherever it's needed in the cells. Right, so this is what's happening in your cells all the time. This is protein synthesis, and it all goes back to how does it know to do this? It's in the DNA. So this is also called the central dogma of biology. Central dogma. Dogma is like a principle or a theory. The central dogma of biology states that information in living things, in your cells, goes from DNA to RNA to protein. Central dogma of biology is the central theory or principle that really guides all processes in living things. States that information goes from DNA to RNA to protein. So DNA is where it's at. DNA starts it. So this is why DNA is so important, because that's the information that contains the information, the instruction manual to make the proteins. If there's mutations, which really are just mistakes in DNA, most of the time mutations are not harmful and they're not helpful. They're harmless and they don't really make much difference. However, there are mutations that are harmful. And if you get a harmful one, it's going to affect everything else. It's going to affect the RNA. It's going to affect the proteins made. So, for example, the hemoglobin, which is a protein, is not made properly. There's mutation that causes sickle cell anemia. So the cells are not even, the red blood cells are deformed, and they can't carry hemoglobin very well. And that causes a lot of problems with people that have this disease. They can't get oxygen. But it's all due to just a mistake in the DNA, which programmed a mistake in the proteins eventually. Okay, so DNA, and that's so important that this is preserved. So damage to the DNA causes cancers. It can cause a lot of different diseases. So this is why the protection of your DNA is very important. And nature knows this. That's why the DNA is in the nucleus of the cells. It's protected. It's like in quarantine. You can't escape. It goes out. People aren't practicing social distancing. Might end up getting diseased. Okay, so DNA, that information, RNA, goes to RNA. And that information from RNA is converted to proteins. So if there can be mistakes throughout the process. But if you start with a mistake here, you're going to definitely have mistakes there. Okay, so the process from DNA to RNA, this process is called transcription. Going from RNA to protein. Remember, in this case, now we're going from one language to another language, going from genetic code language to protein language. So this is translation. Transcription happens in the nucleus. Translation happens in ribosomes. This is the information flows through living things in this direction. Starts in the DNA.